6, 5, 4, 3, 2, 1, 0. We have commit and we have liftoff at 2:13. 7.6 million pounds of thrust, and it has cleared the tower.
Prepare yourself for a world of science.
What is going on, everybody? 10 o'clock on a Saturday, and we are here. The Science Knights are assembled. We have Dr. Thomas Shiller, Dr. Sean Graham, and Dr. Anirban Bhattacharjee here in the studio. They're going to be talking a little bit about CRISPR. And I don't know anything about this. I'm assuming it's something to do with genes and modifying genes, but at first glance, it kind of sounds a little tasty. Why don't you tell us about it?
All right, thanks, Conley, and good morning, West Texas. We are the Science Knights. As Conley said, today we're going to be talking about a topic that's been in the news recently, a really important topic related to a discovery recently made in cellular and molecular biology, CRISPR. Now, what we're going to do today is try to explain what exactly CRISPR is, why it's important, and some of the controversies surrounding CRISPR. But before we get into the juicy details about CRISPR, Dr. Graham, would you mind kind of filling in our audience about what exactly a gene is and what genetic engineering or genetic modification is?
Yeah. So DNA, a lot of people realize what it is, but maybe not exactly. It is kind of a weird topic that people seem to know a lot about, but exactly what it is and what it does is not that clear. So
you also have the RNA.
Yeah. There's different kinds of DNA and RNA are both kinds of nucleotides. They have a certain chemical structure, and they're involved in heredity. So DNA is essentially the way that organisms store information. And the information that's being stored there is basically instructions on how to make proteins. And we mentioned this before on another episode when we talked about genetically modified organisms. So DNA, if you want to think about it, every organism, including you, has this huge library of information. You can think about it as a giant dictionary, right, with millions of pages. And all those little letters in the dictionary are the individual nucleotides. The little individual letters that make up DNA are, you might have seen it before on TV. It's like a little readout that has G, C's, A's, and T's in different combinations. G, C's, A's, and T's. Those are the individual nucleotides of DNA. And what they do is a little three-letter acronym, a GCC or AAA or TTT. Those stand for amino acids. And amino acids are put together to make proteins. So a gene, in that analogy, for your big dictionary with all the information that is there to make you, a gene would be a single page of that dictionary and that page stands for a protein. And again, proteins do everything.
So basically talking about you would have in a gene multiple DNA molecules in there, right?
Yeah, a whole page of letters.
A whole page.
And really, in reality, is something like 40,000 kilobase pairs. So like it's millions of those G's, C's, A's, and T's that make a protein.
Or make a gene.
Yeah.
A gene. And then we also know there's a thing called genome, right?
A genome?
Genome project. So we talk about, and that is mapping out the entire different types of genes a person can have, or an animal.
Right.
This is a huge project that we have taken, right?
Well, it's already done. We finished it back in the early 2000s for the human genome.
We are doing it for
currently people are sequencing genomes of all life on Earth, and we're getting really a lot better at it. It's getting cheaper, and so when the first genomes were being sequenced, figuring out what the exact sequence of all the little G, C's, A's, and T's in a whole organism's, all of its genetic material. One of the first ones we did was a bacteria, E. coli, because that bacteria, and we'll talk about E. coli again, it's a famous study organism that's really easy to grow in a lab, so you want to know everything about it, and people have done stuff with it. So we did E. coli a long time ago, but then when we set out to map the entire human genome, that was a much bigger undertaking. Of course, we're done with that too, and we're getting more and more genomes. And the genome, all the genetic material of an organism is equivalent to that whole book that is you, with all the pages indicating a different gene.
So let me put it this way. Like, if I was to go talk to somebody, and in my head, I've always wanted to visualize this. It seems like you have this huge dictionary. Let's say that's the genome. That's the English language dictionary. That's your genome right there. And then you go to letter A. That might be the gene. Let's say letter A is the gene, and it has all the type of possible combination that the words that will start out with A have, for that matter. And inside that A, you will have now corresponding different words which can now represent your DNAs in there. Would you say that's a good way of saying that? So like a breaking down step by step?
I think I'm not sure if that, because maybe the different letters could indicate different kind of classes of proteins that are being made by the different pages.
Okay.
But I think it's so in weird ways DNA is it's way more complex than we're talking about, because it's not just a dictionary. It would be like a ton of dictionaries. That's how much genetic information is in a human. And at the same time, though, the letters that make the words are more simple. There's only 4 letters that can combine to make 3 what we call codons, or sorry, it's not 3 total. It's there's about 24 different codons. Sorry, there's 24 different amino acids and about 60 codons, which are those 3-letter combinations of A's, G's, C's, and T's that make just 24 amino acids. That's it. And so it's in some ways, like, to try to analogize it to human language, there's differences.
Yeah.
And so our words are way more complex. The number of letters that we can use to make words are more complex. Yet DNA, being more simple in terms of the number of letters and the number of words, which are just 3-letter codons, and the total number of, what they translate to, those little proteins, way more simple. Yet, think of the complexity you can get. And we have about 20,000 genes, or, the numbers vary depending on what textbook you look at. Way fewer genes than would have been expected after the Human Genome Project was done. We're like, wow, there's way fewer genes in a human than we ever expected, given the complexity of humans. But you can do weird things with those genes. You can take a single gene and you can cut it up and turn it into a different kind of protein, depending on how you kind of edit it, how you splice it together, and that's how those kind of things lead to more complexity, and all that leads to a bunch of different kinds of proteins, and that's what the name of the game is. DNA again is good for 2 things. It can serve as a template for its own replication. If you pull it apart and add the corresponding sequence on the other side, it's easy enough to pull DNA apart and replicate it. And the other thing it does is it serves as the code to make proteins.
So I do want to make sure, if for people who do not know what DNA looks like, it is basically a helical structure with 2 sets of molecules basically spiraling one another. And in between that, think of like a ladder which spirals around. That would be a good picture.
It's a twisted ladder. And
so.
And each strand is going in opposite directions from each other. That has directionality. And the really cool thing about it that makes it easy to replicate is that what we call complementary base pairing. So the little nucleotide with its G, C, or T, if there's a G on one side, there's always a C on the other side. And if there's an A on one side, there's always a T on the other side. So if you pull it apart and you have the G and A on one side and you add the C and T on the other, you've essentially replicated the DNA super easy. It's a really elegant way to do it.
So this kind of brings up what we're going to be talking about today with CRISPR. CRISPR, the modification or the use of CRISPR is how we're doing one of these wacky type of genetic engineering or genetic modifications, or potentially we could, and the implications behind that are really kind of critical. It's important that we understand that we have the capabilities of recognizing, identifying, and locating different strands of DNA, what they're for. But with this new discovery, we can actually modify that, right?
Yeah.
And-
We've been able to modify genes for a couple decades now, but it's been kind of slow and it's been kind of hard. For the longest time, we've used what are called restriction enzymes to make cuts in DNA and to try to insert new genetic material into certain organisms. That's how you can make genetically modified organisms. Restriction enzymes will do a double, it'll break both sides of that DNA strand in two places, and then you can kind of cut in whatever material you want, or maybe even to cut out material to introduce a mutation on purpose. And restriction enzymes, the problem with those, or the reason why they're not easy to use, is because they will only cut DNA at a certain place. It actually seeks out a little letter sequence, GCAT, whatever, and it finds that little spot and it cuts DNA there. So we've been kind of limited into where we can cut DNA. It's been really it's been difficult, but we've done a lot of cool things so far. For a while, and most of the kind of cutting we do is in organisms that are really well studied to where we can manage it really easy. So E. coli, fruit flies, and the laboratory mouse. So those have kind of, and we've done great things with just those three organisms. We learned a lot by having limited ability to do editing on a limited number of creatures. CRISPR allows us to do cuts anywhere we want in any genome. That's the game changer. And I know that may not sound that big, but it means that now we can easily edit human genome. It means that we can easily edit anything. If you wanted to study a salamander, there's no problem. From my point of view, being like a guy who studies amphibians and reptiles, you can never do these kind of cool physiological studies like they do on mice. I'll give you an example. We have what are called knockout mice, and we've been doing that for 20 years. You can purposefully introduce a genetic defect into a mouse by doing some cuts, introducing a mutation on purpose, and you do this in a little embryonic little mouse cell, and then you introduce that into a mama mouse, and then she has a little baby that has a genetic disorder. And you do that to study human genetic disorders, right? We don't care about mice. We use it because mice are close enough to humans where you can get, and lo and behold, you introduce a knockout mouse that maybe has some kidney function disorder, and it's exactly like a kidney function disorder in a human that you can't study that way because you can't do experiments on humans. And that's why we make knockout mice.
And I was going to say one thing. And so what we do is, as I'm assuming, that when the scientists look for these genes to edit and stuff, they're usually looking at the genes that are common in both mice and humans, right? That would be the target. You wouldn't just do it on something that is completely as you.
Yeah. Well, sometimes people do these fishing expeditions where they're just kind of screening, looking for whatever they want. But usually it's directed towards something that has human application. So a great example, I always use this one in my classes. There's a great picture of two mice, one of which is about 4 times the size of the other. It's overweight. It's obese. And so the purpose for that experiment, they used a knockout mouse where they knocked out the function of a hormone called leptin, which is involved with satiety, when you feel better after you eat and you're done, you're finished, right? So they messed with leptin in mice and found that they couldn't get these things to stop eating, right, whenever they had leptin disorders. And so they were doing that because they were hoping that this would have applications where they could somehow treat humans with obesity with some sort of leptin, enhancer, and then you'd feel better after only eating a few calories. How amazing would that have been if it worked? It didn't. Yeah, it didn't work.
Of course not.
Turns out the key difference between mice and humans are way better at putting on lots of pounds because in our kind of evolutionary history, we had to go a long time without eating, and when we got back from 2 months on the hunt without getting any food, and we suddenly had a buffet, we would put back on pounds. And that unique ability of humans to have kind of become skinny and that get really big is one of the big problems that makes it impossible for us to work the same way as a mouse. Mice are different.
And also another thing about humans is we have to understand we do have some sort of a higher, I would not put it in that regard, is like abstract thinking comparatively, like even
psychologically, I think it's probably you could probably overcome if you're like, still, I want to eat that pizza even though I feel good, you know.
Yeah, it's the week after Thanksgiving while we're recording this, so we all felt that last week.
Yeah.
One more serving of turkey.
Yeah. So psychologically, there's that complexity might make it different. But the kind of point is, you know, we've had the capacity to do that with mice and other really well-studied organisms, and the whole point of studying things like fruit flies and bacteria and mice is not to know more about fruit flies, it's to know more. It's amazing how many genes humans and fruit flies have in common. You can learn a lot about people from studying fruit flies.
Isn't it like true also, like, sort of butting in, is like banana has like 70% of the genes basically.
I don't know if it's 70, but because they're, you know, we're eukaryotes. We're organisms that have nucleated cells, and if you have nucleated cells, you've got a lot of material for cellular activity in common with other things, even though they don't look anything like you.
And one thing I would like to point out when Sean was talking about is we do this things on E. coli bacteria and stuff like that. So I was looking at this like nice little small video. Any layman with even no knowledge of biology can grow bacteria in their house. Like, you just need a slide, and you can just need.
Oh, you're growing bacteria in your house on your upper lip. It's all over the place.
Yeah, and my nose, and like so. No, I was going to say, like, if for an experimental purpose, like, you can just have an agar plate and have a bacteria on it, and
it's easy. It's easy.
Yeah, it's very easy.
A lot of these kind of DNA cloning techniques and various splicing techniques, introducing new genetic material into bacteria. That's stuff that has been going on for so long and is so easy to do now that like sophomore level students at universities are doing this. And here's the thing that's crazy, let's get back to CRISPR. CRISPR is so easy to use that the same thing's going to happen. In fact, I just met an undergraduate student at a university from New Mexico who told me that he was working with CRISPR.
So, before we get into CRISPR and all those, how much would a machine like which does? I'm assuming you need a setup.
It's not super cheap, but the cost is cheap enough to where it's within the realm of like your ordinary biology lab cost fees, you know, like for a university where they're going to have a certain amount of money to pay for reagents and things like that to do in the lab and all the little vials and the pipettes. It's within the realm of an ordinary university's budget.
So we can easily what you're saying.
We can do it at Sol Ross.
So we can do it at Sol Ross.
Yeah, there's some of these things that we've got the stuff laying around. We could do it at Sol Ross.
So we can just put in a request for this, and hopefully it gets granted.
A little bit more money for I don't know, and I wouldn't know the foggiest thing about how to do CRISPR. But with all these kind of molecular techniques, what it comes down to is it's always like a cookbook. You have these instructions. It's much more precise. So you're not doing that thing where you're like, well, a pinch of salt. It's like, 10.
Not alchemy.
Yeah, it's 10 microliters of this. It's real precise stuff, and you got to keep it clean. You got to, but it's I've done a little of this kind of stuff. With DNA kind of stuff, you want a clean room. You're gonna be wiping.
So by the way, when we talk about a clean room, we are talking about a really, really sterilized room. To the how much we can get it, but so we have to make
any
that
kind of just a small strand of DNA in your sample can contaminate.
And you have to wear hazmat suits and everything and get well. Wouldn't you say that? I would think like if you want,
it's not a hazmat, but it's like close. It's close. Yeah, you'd be wearing. You look like a scientist when you were doing this.
It sounds like Anirban's gearing up to do this in his living room.
Yeah. What is this, Anirban? What are you trying to grow?
Some pipettes.
What are you trying to grow?
And some CRISPR machines.
All right, well, we're going to take a quick commercial break. When we come back, we're going to define CRISPR and talk about why it's so important. We'll see you after the break. Welcome back, West Texas. We are the Science Knights. Today we are talking about genes, and specifically we're talking about this relatively new discovery of CRISPR. So, Dr. Graham, what exactly is CRISPR? What does it stand for, and why is it important?
Yeah, CRISPR is an acronym for Clustered Regularly Interspaced Short Palindromic Repeat. And molecular biology has an acronym problem, guys. It's got a big acronym problem. All these things, it's very complex, and molecular biologists love to create acronyms. And if you read any of the papers in molecular biology, They're what I call a word soup. It'll just be a bunch of TLAs, which are 3-letter acronyms, and then a bunch of FLAs, 4-letter acronyms. And it's just full of acronyms. They're always defined first at the beginning of the paper, and then it just goes away. So you're constantly having to go back to look, and they're a nightmare to read, these papers.
And also, it is not just your field. It is something normal for every field. Is basically, if you're not,
I don't know, man. I think I've read some stuff in other fields, and these papers are practically you can barely read them.
No, I'm just saying, like, even in a normal field with normal usage of acronyms,
acronyms are common in science, but they go into overdrive. So this is an acronym, and what that's referring to, this palindromic repeat. What they're talking, a palindrome is like a word that's the same going one way as it is the other, right? Like ABBA. Mm-hmm. And so when we talked about those GCs, A's, and T's, so if it said TATA, I guess that's not one. ATTA would be a palindrome. Yeah. And so somebody they were discovered, these little palindromic repeats were discovered back in 1987 by people who were working on E. coli, a little bacteria, and they found in parts of its genome, they found these little short repeating palindromes, and they didn't know what they were even for, and they just noted it, wrote a paper about it, said, hey, there are these things, we're going to call them CRISPR, and then it kind of went away. Nobody really knew anything about what they did until people kind of came back later to study them and realized that what essentially they are these little collections of DNA that are saved by bacteria from previous infections with viruses. So viruses can invade bacteria too. We call them bacteriophages. And viruses are just little pieces of genetic material. Often they can be single-stranded RNA, they can be single-stranded DNA, they can be double-stranded DNA. It's just a little piece of genetic material that hijacks a cell's molecular mechanisms to make new pieces of DNA, and then they'll get inside bacteria, use the bacteria's machinery to make copies of itself, and then they have the audacity to leave that bacteria and kill it on the way out. If they didn't kill the host cell, it probably wouldn't be that bad, and maybe we'd never even know what DNA were, what viruses were. So anyway, some bacteria will actually save little pieces of the DNA that have previously invaded them, so that it's almost like an adaptive immune response. They'll cut the DNA up with these enzymes, and the part of CRISPR that's called CRISPR-Cas9, that Cas9 thing is the enzyme that can actually make the cut to DNA. And it'll cleave DNA at a certain point, and those little repeats and their associated genetic material that are supposed to actually figure out a part of the bacteriophage that is specific to that bacteria so that it can cleave a virus that has invaded it before. So it basically remembers when a virus has come back.
So let's take one little step back. So Cas9 is an enzyme.
Yeah.
And enzymes are basically, you would say, how would you...
An enzyme is just a protein. that basically shortens the amount of time a chemical reaction
can occur.
And so there's all kinds of chemical reactions in life forms that are very unfavorable, that don't want to move forward, that enzymes will create conditions to make them go forward quickly. And that's one of the most amazing things that proteins do, is act as enzymes. And so what this does that's unfavorable, DNA, once it's bound together in those nice long strands, it's really hard to break it. The bonds between each individual nucleotide are very strong, and it's very favorable to stay put once you're built. This thing will just whack it totally in half, which is very unfavorable chemically, but it'll do it. It'll do a double-stranded break. It's important to note that when your own cells, copy the DNA strands, that never happens. You never just go into DNA and cut it midway. You unfurl it using these enzymes and do it in this more kind of delicate fashion. It's never like a double-stranded break. These enzymes that bacteria have can do that. The restriction enzymes that I talked about earlier, and then Cas9 can do it. And these little palindromic repeats direct enzymes like Cas9 to go to a certain point at the genome. And so the people who came up with the CRISPR-Cas9 kind of enzyme package said, holy crap, this means that all we have to do is guide the Cas9 enzyme to exactly where we want it to go. using an artificial RNA drive, like to direct it, and we can cut DNA anywhere. And that's what the CRISPR-Cas9 revolution is going to be based on.
So let me get this straight. So now at this point, we don't have to cut it at the points where the pattern is repeating itself, and we can just cut it anywhere. Pretty much, it doesn't matter.
So it's based, the little palindromic repeats and the little piece of DNA is saved from previous infections from viruses, just the way that bacteria can direct a cut to cleave the DNA of a virus. So now we can make little artificial stretches of RNA that will go and find its corresponding area in the DNA to cut, right? So we've just harnessed this little tool that bacteria use to cut up DNA, and we can use it to cut up DNA, and we can further put in the package a new gene to insert. So we can cut out a part of the DNA and insert in whatever we want. And the cells, this is great. One of the best parts of this, you don't need additional enzymes to fuse it back together. Your cells will do that immediately. As soon as you cut it out and add something in, it'll come in and repair it, and you don't need to do anything else. The cell will do that itself. It's amazing. And so what this amounts to is a technical way to edit DNA any way you want.
So what does that mean for us? That means we can eliminate genetic disorders.
Illnesses. We're just now at the cusp of what it means for us, and I'll give you some ideas about the kinds of things that we can do. First thing, that stuff we talked about before the break about making knockout mice and introducing mutations on purpose to see what will happen, studying organisms that way. That's gotten about 100 times more easy now. And you can study it in any organism, not just mice, fruit flies, and worms. You can do those kind of cuts if you want to in human cell cultures, right? Cultured cells, human embryos if you wanted to. That's actually been done, unbelievably. And cancer, at its heart, is a genetic disorder. And so if the only problem now, we can already, I guarantee you right now, they could take a cancer cell and they can fix it using CRISPR. The only trouble now is how do you get CRISPR to all of the cancer cells to fix them? That's just a technical problem at this part. And once they overcome that technical hurdle, we've done it. And cancer's gone.
So I want to hold on to that a little bit later. Maybe we will discuss this about the more advantages that CRISPR will bring in and all those stuff. But I would also want to talk about a little bit. So how is CRISPR now different from, is CRISPR then the same as what we typically hear in the news about gene therapy and stuff?
So CRISPR gene therapy.
What is
gene therapy? Has been, we've been working on that for a while too. And this is the idea that with a genetic disorder, or cancer, all it is a faulty gene. And so if there's a way that you can go in there and fix the gene, that's gene therapy. Now, the only thing that we have in our toolkit right now, there's, or what we did have before CRISPR, to deliver a good gene, right, is we have viruses that can do that. We can basically make viruses go in and fix genes because that's what viruses are good at. They've got some of the enzymes necessary to open up the DNA and insert in itself. And if you can kind of train viruses to put in a repair gene, then you're off to the races. The problem is, the genome has that same disorder if you're born with it, right? If it's a genetic thing that you inherited, all your cells have it. So how do you get a virus to invade all of your cells and fix it? So that's a technical problem too. If we can ever overcome that technical hurdle, we've cured all genetic disorders, and they're working on that. With CRISPR, it's just made it a lot easier. Making the little cuts, putting in a repaired gene, all that stuff's got 100% easier. There's a quote from a paper I was reading where it says, for cancer research, many of the challenges in generating germline or somatic mutations have become trivial, unquote, because of CRISPR. And this might be a good time, they're talking about germline versus somatic cells. Most of your body cells are ordinary cells like hair cells, cells inside your gut. Those are what we call somatic cells, body cells. They have 2 pairs of chromosomes. And if a mutation happens to a somatic cell, nothing might happen at all. But somatic cells are the ones that essentially, if they're changed through various mutations, you can get cancer, right? A skin cell can, it's ordinary genes that are responsible for duplicating itself and growing. Those become faulty during a mutation during your lifetime, and then they start going haywire and they just do their own thing. The germline are those that are actually, for making gametes, for making sperm and egg cells. If a mutation happens to the germline, it can actually be passed on across generations, and you can be born with a mutation that maybe make it easier for you to get cancer. So it turns out the easiest way to fix genetic disorders will be the one that's also most ethically challenging or dubious. Because if you want to fix genetic disorders, the thing to do would be to take an embryo after it's fertilized, use CRISPR to make cuts and edits, and then introduce it back into mom.
And talking about that, so you said every cell has chromosomes, and chromosomes in relationship to DNA, how would you...
Yeah, so chromosomes are, basically just tightly wound DNA.
Okay.
Chromosomes also have protein, so there's a little bit of protein, a little bit of DNA, but it's actually mostly protein, but it's just a bunch of DNA wrapped around proteins into these thick little sausages, basically, that when a cell divides, they have to be packaged.
So basically, it's just a DNA in another configuration. So
yeah, think about chromosomes as just like there's so much DNA in your cells that if you look under a microscope when a cell's about to divide, you can actually see the DNA bound into those little sausage-shaped chromosomes because there's so much of it there.
Okay,
yeah.
So I just wanted to make sure people have an idea. So we have now on this stage we are ready to go into CRISPR into more detail about where we can apply them and stuff like that. But even before we go, I want to come back to this thing about this germ cells and germline and somatic cells, right? And these mutations that we talked about. So basically what we are saying in this is when the cells are reproducing, there's a problem with that replication, and thus
that's one way to get a mutation.
Out.
If the cell's dividing, and it's got to copy all that DNA before it divides. So the cell that results has all the DNA too, and so a little mistake can be made. Just one of those little G's could be replaced with a C or an A with a T or vice versa, and that's all it takes. That's called a point mutation, where it's just a single base pair change, and that's all it takes. Or it can be big stuff like whole segments of chromosomes attached to another. It can be big, it can be small, but that's all a mutation is. It's just a chance difference in the genetic code.
And this brings us back to like the thing about enzymes and bonds being very strong. What we have to remember is one of the things is bonds break because you have to give them energy. So if you provide things with sufficient enough energy to chemicals inside your body, they will break and they will create weird things. And that is why being in a radioactive environment is where you have really highly energetic particles.
Yeah.
That's where the problem comes in. They interact with your molecules in your body and they split them up in a ways which you don't want them to split them up. That's
radioactive material, carcinogens, chemicals. There's ways of inducing mutations, but of course a mutation, it doesn't have to be induced. You could have cancer and not ever have come in contact with a carcinogen, or it might not have resulted from contact with a carcinogen. It would just be a chance
no, I'm not saying that carcinogens, but I'm saying like with radioactivity being in a nuclear reactor without being shielded,
increases your chances.
It is pretty much you are on a 99% probability. If you're not shielded up, you're going to get cancer.
Yeah.
So yes. So and so this is how we get this, and sometimes and there are other things we have, and now bring it back to genetic disorders which are transmitted through humans, like from mom and dad. Like one of the common things we do know is hemophilia. That would be a genetic disorder. That is not a
right. It's a mutation. Your clotting doesn't work. Yeah, there's lots of things like that.
And another not a genetic would be insulin, like for diabetes. Would you say that is genetic or not? There's
definitely genetic predispositions for that kind of thing.
So it's obvious how useful this discovery could potentially be in the future, but when we come back from the break, we're going to talk a little bit about some of the controversy behind it, because obviously there is some potential controversy. All right, welcome back. We are the Science Knights. Today we're talking about CRISPR. Now, before we get into some of the controversial topics related to CRISPR and genetic modification and all that, let's talk about some of the good things that could come out of this discovery.
Yeah, thanks, Thomas. The main things, I'll give you guys kind of a slice of what researchers are currently using CRISPR for right now. This is kind of the cutting edge. One of the things is creating, knockouts. It's really easy now, knocking out a gene. So you can put a bunch of cell cultures of various cells in a bunch of multi-row plate, and use a computer to design specific knockouts of specific genes, and then run them through a screen and kind of determine what function has been lost. And this is just a great way for research to study function in cells. And that's something they're already working on. And you can do this like thousands of genes at a time because of CRISPR and because of computers. For cancer, University of Pennsylvania has gotten approval from the FDA to use CRISPR to modify white blood cells from patients to attack cancer. So they're white blood cells. Your white blood cells already, if they're good, they already go after cancer. They can tell a faulty cell of your own cell. It's hard to do because those cancer cells, after all, are your cells. So that's one of the ways that cancer is pretty bad, is because it can evade your immune system. But your immune system can locate cancer cells and kill them, and it does all the time. I guarantee most of our listeners here have had faulty cells harvested and killed by your own immune system so far. But sometimes they don't notice those cells because they're, for whatever reason. So the University of Pennsylvania harvested white blood cells from patients with cancer. They knocked out 3 genes that were preventing them from noticing or from proliferating fast enough to control the cancer, and they showed varying results. It was only a study of like 3 or 4 patients. But that's a great, like, training your own immune system using CRISPR, maybe developing antibodies using CRISPR that can recognize cancer. That's a way of doing this without having to do any kind of really hardcore modification of the host. All you're doing is taking the patient's white blood cells, modifying them, putting back into the same patient, right? So you avoid all of that kind of ethical, scary stuff. Gene therapy. Trying to use CRISPR to modify genes of people who have genetic disorders, and what the problem with that is just the delivery system. How do you get that CRISPR and the new gene into all those cells? There's ways to do that with harmless viruses. There's ways to do that with lipid nanoparticles. There's ways to do that with this technique called electroporation, where you're just kind of shaking up the cells using an electric current to introduce the CRISPR package, right? So you're not even using a virus. This works real well for if you have a genetic disorder that's only affecting
Localized.
a local tissue. If you wanted to try to fix somebody's pancreas and change the genes in just the pancreas or the eyeball or something like that, this works. If it's a genetic disorder that's affecting all the cells in the body, it's going to be hard because you got to deliver that gene to all the cells in the body. But if you just want to do a local fix, CRISPR is going to be a game changer for that.
And one way we can, one of the things that we can think of as not a local is hemophilia, which affects the entire body. So that would be hard. But what I think with CRISPR, what you can do is treat the parents from being, or rather, in this case, hemophilia, it would be the mother.
I think the point where you'd want to do it, if you're trying to fix a genetic disorder like that, and this is where people are going to get real squeamish. The embryo, when it's just 2 cells or 1 cell. And, this would be kind of like, a test tube baby, right? You would donate your egg, you do in vitro fertilization, and you could do that with real mom and dad, right?
And that's been around for a long time.
We've been doing that forever.
Yeah.
And then you got that just 1 cell now. And then you use CRISPR when it's just 1 cell to fix the genetic disorder and then put the cell back into mom. And let me tell the listeners right now, nobody's talking about doing that yet because we don't know what the implications would be, although I got to back off that because a Chinese scientist did do that. But nobody here in the U.S. is talking about doing that. I'll also mention to the listeners that China is all over this. Like they've made leaps and bounds with AI technology. They're doing the same thing with CRISPR, and they're being very secretive about what they're doing. They're not publishing all their results. They saw what happened when their scientists did a fix to an embryo, and it made everybody panic, and it caused a worldwide sensation and of condemnation. And so now they're just being super secretive about what they're up to.
Let's hold on to that one, the ethics of that. Let's talk more about the good things about CRISPR, and it can help. For example, we can make our plants even more bug resistant for that matter.
For all the things, if you're comfortable with genetically modified organisms, then CRISPR is just going to make all that so much easier and cheaper.
Yeah.
All the things that we can do using the power of genes to introduce, plants that can uptake nutrients quicker, and maybe reduce our pesticide use by using genetic modifications to kill mosquitoes and that sort of thing. All that stuff's going to get easier. And just one more medically significant one. One of the major cancers and many problems that you can get that are genetic in origin are problems with your blood cells. Lymphoma, leukemia. Those are genetic disorders, cancers in your bone marrow cells. Well, the cool thing about that, then it becomes localized, and bone marrow cells are stem cells. They can become different kinds. And so what we were working on right now, they're working on therapy for sickle cell anemia, because that at its heart is a genetic disorder to red blood cells, and they're formed in the bone marrow. And then white blood cells that lead to lymphoma, leukemia. There, we're already doing kinds of things where we do bone marrow transplants and that sort of thing. And the thing what you do there is you just kind of take one healthy cell out of the bone marrow of a patient, or you get a donated blood cell from someone who's a relative, and then you bombard that patient with chemicals or radioactivity that can kill all the bone marrow cells. And then you replace the bone marrow with healthy cells, and then it grows back really quickly. And then it reestablishes their immune system and their red blood cell system. Now, you could use CRISPR to modify the patient's bone marrow cell to fix sickle cell, introduce it back into the patient, and they're cured of sickle cell anemia. Cured.
Or basically do another thing. I was thinking, so the stem cell that we are talking about is also like the T cells and stuff like that, right?
Yeah, all the really cool immune cells you've ever heard about, B cells that make antibodies, T cells, T helper cells, all those are made in the bone marrow. So if there's any genetic disorder to any of those kind of cells, I think in just a few years we're going to be able to fix all of that.
And what we can do is basically, instead of not even have to replace it, what we can do is go to the part of the so, quick question. Is the bone marrow, has it been produced entirely throughout, or has it been controlled by, of course, brain controls everything. Can we instead do something at that part and then that it in itself will just produce that type of
I
healthy...
think you could probably try to do some targeted CRISPR editing in the bone marrow itself, the bone marrow cells. But the reason why we do those really kind of overpoweringly troubling things like, bombarding people with chemicals for treating cancers, because you want to make sure all the defective cells are killed.
Gone, okay.
And that's hard to do because you got to be in the bone marrow. So the way they do it, they just kind of clean the slate using chemicals or radioactivity. It's tough. The patient has a hard time. You lose your hair. You lose a lot of your fast-growing cells, but then you can recover from that. And if they get every last one, it just takes a single bone marrow stem cell to replace all of those cells. And if you replace it with one that's been fixed with CRISPR, it's the patient's actual cell, and it will reestablish itself, and that's wonderful. This is a way. It's a really good thing, especially for that kind of a local effect. And like I said, this is something people are working on. It's FDA approved. They're working on it right now. This could be, within 10 years, we could be seeing leukemia, lymphoma, sickle cell anemia, all of that stuff. Anything that has to do with bone marrow cells. We have a high chance
of getting that solved.
We got a really high probability that all that stuff's going to go away.
In a much less painful way. Because, and the second thing I was going to talk about, more fun side about now, let's talk about. So we all know there are life forms that are, we call them as extremophiles, which survive in extremely extreme conditions. And and there are bacteria and stuff which will survive at over 100 degrees Celsius, which is a temperature of boiling water. They can survive in water which is like extremely salty. They can survive in water which is extremely cold, where we would not do. So they push their enzymes and the protein enzymes to function at a certain level where our normal human body or normal animals would not do that. So can CRISPR take those things and put it in our human body so that we can walk around in shorts in Antarctica?
The short answer is yes, pretty much. Yeah, any kind of genetic modification you want to make. We've already been working on genetically modifying extremophiles to do things that we want them to do. So that's been going on a long time, but now it's just easier and cheaper because of CRISPR.
Sure.
But yeah, doing some kind of recombinant thing where you give people special powers of X-Men, of course, then you're going to run into ethical problem, and that's what we should talk about now.
That's what I was
talking about
the
trouble with CRISPR, any kind of ethical. I think the big thing, the Chinese scientists that did do a germline modification to a human embryo, that was a big deal because nobody really, he didn't think about the consequences. If you knock out, and what he did, essentially, I think if I can recall, he knocked out a gene for T helper cells that it makes some sort of a surface receptor for T helper cells that allows HIV to invade T helper cells. And that's what HIV does. It invades them, kills them, and after several generations of killing T helper cells, your immune system can't function anymore, and you get all these opportunistic infections, acquired immunodeficiency syndrome, right? So he didn't think about what other problems those T helper cells could have with that knockout of function. And so the baby that was born from that experiment, people will be watching it closely, I'm sure. But that is that's the main kind of hurdle right now with any kind of human modification is you got to watch out for this kind of domino effect, the snowball effect. What other problems could you introduce by making these kind of cuts? Unintended consequences. So that's one of the big things. And that's why it's going to be a long time in our country before you see, it's going to go through lots of approval. FDA is going to have to sign off on it. It's going to take a long time before this kind of gets caught up with to where we realize that, when we do this change, it's going to be a good, healthy change.
I would like to point out that I think this just came out, and I read this yesterday night. So this is just one simple paragraph about the Chinese scientist who did that. I'm going to read this out from Guardian, like word for word. The gene editing performed on the Chinese twins. So it was twins, not like
right.
Just
That's
single.
Cool. So you could. Did he do it in both the twins?
Yes, both.
Okay. I would have thought he would do it in one, so that he could see.
I think he did it in both.
So he could have a control. He could have a control. Well.
So twins to immunize them against HIV may have failed and created unintended mutations. Scientists have said that after the original research was made public for the first time. And I was reading through it, is very, very scary that the person who did they at the end of the article, it said authors also appeared to have taken steps to make it hard to find the family, like leaving the names of the fertility doctors off the paper and including a false date of birth. So,
well, that's kind of par for the course, though, because a lot of times for privacy purposes, you never know who any of the patients for any of these studies are, and that makes sense. But it's troubling, and so that's one of the big. That is amazing that as soon as CRISPR kind of really hit the world stage, immediately a controversy that kind of demonstrates the ethical implications was there. The other stuff, the other pitfalls of CRISPR, kind of mostly technical. How do you deliver the new gene into cells in a human body? What kind of off-target effects? So you're trying to affect one target. Would you introduce this gene? Are there domino effect that go after other kinds of things? Other problems. This is kind of a good one. If you try to introduce CRISPR, your immune system will typically notice it. It's a big fat enzyme, Cas9. They're big proteins, and so your immune system's going to recognize that as not your own, and it's going to tackle it. So trying to use CRISPR in the context of a human body where your immune system is going to try to attack it, Those are all technical problems, though, and that most of that kind of stuff with using CRISPR, where a lot of scientists are saying, "Hey, wait a second, this isn't going to be the cure for cancer necessarily." It's all because there's still a lot of technical issues involved in making it work.
So, and basically, we just want to make one thing clear before we start demonizing the parents and everybody. It's one thing we do have to remember, that if you are a parent and if you know your child is going to have a genetic disorder and you have only one shot, and there's only one shot having a child, would you not take the risk? Because these are the questions we before we start demonizing everybody, it's very hard to, because if you know your kid is going to have a genetic disorder coming out and CRISPR can treat it, what you do is, how would you play it now?
My guess is, this is a classic kind of conundrum. It's like it's real easy for us to say sitting here right now, oh, we better watch out because you're not the one who has a 95% chance of inheriting this genetic disorder or living with it for your whole life. So I think that in the future, that's where we're going to be, is that we're going to work out the kinks. We're going to work out a lot of these technical problems. And in 50 years, people will be like, wow, can you believe that we once lived in a world without CRISPR?
And where we could not walk around in shorts in Antarctica.
Right.
Yes, that's
exactly.
That's the point.
Because of the gene put into us from a extremophile. I like that.
Yeah.
And hey, we hope that you enjoyed this show, and tune in next week as well, because as always, we're going to have great new science topics for you to listen to. Good night, everybody. Good morning, whatever time it is. I always forget. We're the Science Knights.
Thanks for listening to this episode of Science Nights in the Morning. Be sure and follow us on Patreon for exclusive gear and uncut episodes. Check out the latest science articles on our Facebook page and subscribe to us on YouTube and your favorite podcast listening app. You can also listen every Saturday at 10 a.m. Central Standard Time at BigBenRadio.com. And if you got a question, we'll join the discussion. Hit the hotline at 432-217-1983 and record your message. We couldn't do this without you, and thank you so much for listening each and every week. That's Science Nights in the Morning with a K, and we'll see you next time.